Thank you, everybody. Thank you, Claudia, for the introduction. And as I mentioned, we are a company, an Italian company based in Bologna, and we have two branches in the southern part of Italy, in Sicily and Sardinia, the two islands. What we do is we create new technologies for the biotech industry in order to help researchers create new data and results on their 3D biological samples like spheroids and organoids. We found a gap related to a biophysical characterization, and actually talking with many researchers worldwide, we noticed that this might be very useful for them. And for this technology, we went to US last month at SLAS 2022, as we've been selected for the New Product Award. In that expo, it was a very interesting experience. And this instrument actually, we launched it is in 2021, on February. And I don't know if you are involved in the 3D cell culture, but there is a recent paper that it's been published in Nature Methods, where top of the state-of-the-art researchers are dealing together to create some kind of database on these samples, and they call it Miss Sphero ID. And three of them are already our customers, or they tested the instrument, so the interest is definitely growing. Very quickly, this is our network worldwide. And how this all started. This started from trying to understand biological relevance of biophysics. And this is a common, this is the common view that researchers have when they are preparing their 3D samples, in this case, spheroids. So what you can see here is a certain number of cells per well over time. So you can see that when samples like these are growing, you might have an information about the diameter to understand how this growing process is going. And all the researchers are evaluating this compaction phase around day 2 and day 3. But what really happens later? If you're only dealing with the evaluation of the diameter, you encounter a plateau over time. But if you see the darkness variation of this bright field imaging, you clearly see that something is happening into the cross-section of the sample. And all this information might be related to several important factors like proliferation, or perhaps necrosis, drug permeation, and oxygen gradients, ATP contents, and all these details. So, as I mentioned earlier, talking to researchers worldwide, we evaluated that the need for a small, user-friendly and plug-and-play instrument for gather more information than just the diameter, and can be really crucial to use it since the beginning of the workflow, but also as milestone to proceed further with their research. And that's why we created this very small and user-friendly instrument, that it's a flow-based gravimetric technology that is combined with an automated sorting by the biophysical parameters. So the also importance of the instrument is to allow the researcher to select their range of the biophysical parameters. What biophysical parameters? Well, all of this in details is published in 2020, where we got the cover on Micro Machine, if you want to check for more information about how it works. But the weight at a glance gives the outputs of size, obviously, but the biophysical characterization is related to mass density and weight. And the important things for the researcher is that they can create sorting, basically evaluating the population to separate by size or by mass density. All of this can be done label-free, no fluorophores needed, in sterile condition, and most importantly, with preserved viability, meaning you can technically replate your samples and go ahead with the procedures. We give the output with a normal Excel file or any similar software, which is a distribution of the population based on the box and whiskers um, displaying of the data. The reliability is very high in terms of accuracy and precision. We validated this with microbits similar to other techniques. And the range of samples we are operating is between 50 and 500 microns. So, as I mentioned, we launched the instrument last year on February, but we already used it for many applications, and this is what I would like to focus the interest of this conversation today. This can range from the biophysical heterogeneity and quality control analysis to the evaluation of the morphological changes in terms of biophysical output. So I would like to start telling you some stories, let me say. And I would like to start with this publication we released on Frontiers Immunology 2020. In this case, we used the weight physical cytometer several times during the workflow. And we started from selecting the right samples to be used. In this case, we started with 4 colorectal cancer cell lines to be analyzed, and we compared multi-parametric analysis to actually create a comparison between biological event and biophysical results. Clearly, the physical cytometer, the viability assay test, which is the cell titer glow by Promega, and confocal imaging analysis. Obviously, we had advantages and disadvantages for all the techniques, but if you're thinking about the weight physical cytometer, we have size, mass density, and weight as output. It's label-free, preserved viability, and it's an operation that you can do in terms of 40 minutes to 1 hour to get a certain number of population that it's representative. Obviously, you have compromised viability for the samples in terms of the viability test. It's an endpoint. It takes around 1 hour, and you probably know better than me about confocal microscopy. It gives incredible results, but it's tough, it's expensive, it's an endpoint analysis, compromised viability, and so on. So here you see this arrow that highlights the workflow of the publication and where we started with the physical cytometer. So we started from the 4 cell lines to evaluate. Same seeding number, same day of analysis, and you can already see from the bright field that among those 4 different CRC cell lines, HT29 appears a little bit irregular, not with a rough shape with respect to the others. In this case, SW620, DLD1, and HCT15. We run the experiment with our system, and we are displaying the weight, the diameter, and the mass density. The first thing that we can notice here is that it's all right, it is correct. HT29 have different parameters, but we can quantify how much. Particularly, what we see is that in terms of mass density, the dispersion of the population of the data is quite relevant. Meaning, if you're starting to select the right cell line to go ahead with your procedures, these might be not ideal because you're already introducing a bias with the diversity of the results. So we started using the physical cytometer to highlight the biophysical heterogeneity. Then we proceed with confocal imaging. We did confocal imaging for all the 4 cell lines, and we performed the output in terms of number of cells per millimeter square, and as mentioned at the beginning, ATP content to evaluate the viability. If we compare these results with the mass density, also in this case, we see how similar is the trend. And we can say that for SW620, we have the higher number of cells per millimeters, higher viability, and higher mass density. So we are starting using, again, the weight physical cytometer to say that there is a proportion there is direct with ATP content in number of cells. But obviously, let's say, we are preserving the viability of our samples. If we bear in mind what it was the mass density data here, then we go ahead with the second part of the paper in which we actually faced the goal, which was try to predict or evaluate whether it could be interesting to see what's the effect of NK cells penetrating into these samples. So we selected the two with the highest mass density, and we went with proceeding with different treatments. Basically, we tested the NK cells at 6 hours and at 24 hours for those two cell lines. And here we can see how the weight is decreasing as well as the diameter for both the cell lines. This is already a very interesting marker to understand what is happening. But things will be more interesting when we are evaluating the mass density. If you can see, there is an increase in mass density. Although we have a decreasing in all the other information, the mass density increases. And this is very interesting because that it's a ratio with the volume. So what we do, we ask ourselves our question though, how come we have something that is statistically significant in terms of differences for the DLD1 cell line, but is not for the SW620? So we ask ourselves some questions like, is mass density a data to predict the infiltration rate? Is this depending from the nature of the cells? So we went again comparing confocal. In this case, we did Z analysis with two cell lines, and we labeled the NK cells with a fluorophore. What it's very interesting to see here is the correlation. In fact, remembering the mass density graph with the higher mass density for SW620 and lower for DLD1, we see that this is directly correlated with compaction. Basically, the permeation of the NK cells would be more difficult. In fact, in the confocal microscopy, they tend to stay on the surrounding edges of the samples with respect to DLD1, where they can penetrate completely. This is something really interesting to evaluate and to correlate with compaction. It is also demonstrated in 2014 by a paper in which they created a process and they tested this Alamar Blue assay on some CRC cell line. And this is to connect the concept of compactness. In this Alamar Blue assay, the higher the fluorescence, the larger is the number of the living cells. So the researchers, they tested two different cell lines and they treated them, obviously standard condition of growth. They treated them with staurosporine, which is a killing agent that is based on induced apoptosis, 1 micromolar. What you see from the output of fluorescence is you have a control and you have a certain intensity, and obviously you're killing cells and the intensity decreases. And that is in line with what is expected. But those are loose spheroids. When they did the same treatment with more compact spheroids, DLD1 and HT29, in the same conditions, the output was a certain number of output in fluorescence and 70 or 60 times higher output after the treatment, which is clearly not in line with what is expected. And this is a bias introduced with the effect of compactness. So what is very important to understand here is that mass density can be really correlated or can really give this information. And in the middle of the workflow, you can evaluate whether your process is going towards the direction that you expected or create some multiple checkpoints. We also published a second paper that goes with this flow related to the permeation and the drug efficacy. In this case, we compare different cell lines, the CRC Lovell cell lines, again, with the weight, confocal microscopy, and protein quantification. What it was very interesting to evaluate is the relationship between the biophysical data that we have from our analytical instrument in terms of 1 hour or so with confocal measurements. So in this case, 7 days of culture, the test was exposed to crizotinib at 24 hours. Here is the imaging in terms of either bright field and more advanced image analysis. If you're only checking the diameter, we have a control, we have a certain concentration, 250 nanomolar of crizotinib, and the double, 500 nanomolar, you have a decrease in diameter. So we performed confocal microscopy and we counted the nuclei, then we calculated the volume and create the ratio similarly to mass density in order to have a nuclear density. What is very interesting is that when we compare live spheroids with fixed spheroids, we have a very, very high correlation of the results in terms of the trend for the treated and the control. So mass density proved to be a quantitative marker from the action of crizotinib. And with the same flow, with the same concept, this has been also demonstrated very recently with another application of our instrument in 2022, where we tested a very well-known cancer agent, which is the 5-fluorouracil with Manuka honey in terms of nutraceutics. In this case, the Professor Cianciosi et al., they tested and evaluated the comparison between the trend of mass density, weight, and diameters among different concentration of only the honey, only the fluorouracil, or a mixture of both of them. Obviously, we need some comparison to understand, and that goes a little bit more into the biology with some cytofluorimetric tests and the markers, which can highlight in blue the live cells, in green the cells that are starting the apoptosis, and in red the apoptotic, the dead cells from the apoptotic process. And what you can see here that with respect to the control in green, the pink variation, which is related to the highest concentration of honey and 5-fluorouracil, is exactly in line with what expected, what obtained from the cytofluorimetric test. So the highest, that said, the highest also apoptotic induced process in green, and the lowest live cells. So also in this case, mass density and weight loss, also with the diameter shrinkage, can be comparable with cytofluorimetric test, but obviously way simpler in order to obtain. I will give you an insight about something that it's on the pipeline but not published yet. This is an example on organoids, and clearly let understand why, if you are only dealing with the diameter, we might miss some information. Prostate cancer organoids, we have to thank the group of the University of Bern in Switzerland for that. They are hollow. So clearly, if you're only dealing with the diameter, there are information missing. And it's been proved that the tumor grows, like the tumorogenesis, can be monitored with our system in order to evaluate at which stage the tumor might grow, because this starts to filling the cavity inside and then increases the size of the single organoids. So in order to conclude, this system also other than being automated, a very user-friendly, allows the biophysical characterization to highlight heterogeneity of samples prior your workflow, but keeping them viable, and allowing the sorting if you need a subpopulation. It also represents the parameters to evaluate viability of your samples and the number of cells if you're interested in that. It gives you insight about the permeation rate, the filtration for both cell lines like NK cells, and also correlation with the anti-tumor drug efficacy. And it highlights morphological changes of your samples. These are the proven conclusion, and many more is in the pipeline. But what we want to highlight is the correlation of the biophysical relevance with biological relevance of biophysical properties. And I would like to thank you for your time. I hope I stayed in the 20 minutes or so, and I'm open to any question if you want.